How do genes differentiate muscular dystrophy subtypes? Imagine your muscles as a well-built house, with each part playing a crucial role to keep everything stable and functioning. Now, think of genes as the blueprints that tell your body how to build and maintain this house. When these blueprints have errors or mutations, the structure can weaken, leading to muscular dystrophy. But not all blueprints are the same, and that's how different types of muscular dystrophy develop. Most muscular dystrophies are caused by mutations in specific genes that produce proteins vital for muscle health. These mutations follow different inheritance patterns, which means they can be passed down in various ways. For example, some types like Duchenne and Becker muscular dystrophies are inherited through the X chromosome, meaning they mostly affect males because the gene involved is on the X chromosome. In Duchenne muscular dystrophy, the mutation causes a complete loss of the protein dystrophin, leading to severe muscle weakness that starts early in childhood. Becker muscular dystrophy is similar but less severe because the mutation allows for some dystrophin to be made. Other types, like limb-girdle muscular dystrophies, are inherited in autosomal patterns, which means the mutations can be dominant or recessive. In autosomal dominant forms, only one mutated gene copy is enough to cause the disease, while in recessive forms, both copies need to be mutated. These subtypes involve mutations in a variety of genes that encode proteins such as sarcoglycans, calpain-3, or dysferlin. Each of these proteins plays a role in maintaining the muscle cell's structure and function. When they are defective, muscles weaken over time, but the age of onset and severity can vary widely depending on the specific gene affected. Congenital muscular dystrophies are another group that begins early in life. They involve mutations in genes related to the extracellular matrix or proteins that connect muscle fibers to their surroundings. For example, mutations in the gene for laminin or collagen can cause these early onset forms, which often also involve issues with the brain or eyes because these proteins are important in other tissues as well. There are also conditions like Emery-Dreifuss muscular dystrophy, which involve mutations in genes that affect the nuclear envelope, the protective layer around the cell's nucleus. Mutations in the EMD gene on the X chromosome or the LMNA gene on chromosome 1 lead to problems with muscle contractures, weakness, and sometimes heart issues. The key point is that the specific gene mutated determines the protein that is affected, which in turn influences how the disease manifests. For example, if the mutation affects dystrophin, the muscle fibers become fragile and break down easily. If it affects nuclear envelope proteins, the disease might involve problems with the cell's nucleus and lead to different symptoms. Understanding these genetic differences helps doctors diagnose the exact type of muscular dystrophy, predict how it might progress, and offer better guidance for management and future therapies. Genetic testing is essential because it pinpoints the exact mutation involved, allowing for precise classification of the subtype. This knowledge also helps families understand inheritance patterns and risks for future children. As research advances, understanding how specific genes cause these different forms of muscular dystrophy opens the door for developing targeted treatments that address the underlying genetic causes. Remember, each person's experience with muscular dystrophy can be unique, so always consult healthcare professionals for personalized advice and care.